All right, welcome. And we are starting our next series in our transfer of energy unit, and we are looking at cellular respiration, or how do we convert that stored chemical energy in glucose and be able to transfer it to usable cellular energy of ATP. So, first, we kind of have to ask ourselves, what is the definition of cellular respiration? And it is the process of breaking down food. The primary one, the one that generally cells go to break down for this, would be glucose. Now, if you kind of take a look, here we have our reactants. In this case, we have our glucose and oxygen, and our products are going to be water and CO2. But take note that we end up getting ATP energy out of this. So we're going to talk about what is the process that happens in this area right here that allows for the power stored in glucose and be made into the ATP. One thing to kind of note, and you probably notice, is that, well, the equation for photosynthesis is just the opposite of cell respiration. In other words, the products from one is the reactants of the other. And so we see this cycle and see how they kind of fall into each other. Now, I will say, when we talk about in terms of photosynthesis, this is where we're putting energy into the system. So we are bringing in sunlight energy from the sun, and we are converting it into an actual usable chemical energy that life on Earth can actually use. And so our cellular respiration then was the process of bringing energy out of the system. And then that way we get it in terms of ATP and heat. And with photosynthesis, the end goal is to make that glucose molecule. So, again, just like I said, the products of photosynthesis are the reactants for cellular respiration and vice versa. Now, let's talk about the ATP molecule. So, ATP is considered to be the cell's energy. Everything that you do consciously, unconsciously, is all driven by this ATP energy. Without the ATP energy, you cannot have life. And so much of our cellular respiration is the end goal to convert ADP, adenosine diphosphate, into ATP, adenosine triphosphate. Now remember, ATP is a nucleic acid. It has the adenine, it has the sugar, but it has these phosphate groups, and that's where the power is being stored at. In particular, in my ATP molecule, the power is being stored in this bond between the second and the third phosphates. And when it's in full charge, it has this energy just built in, ready to go, and to do whatever cellular activity that you need. That energy is released when that phosphate is broken off, and that energy is released and able to do work. All right. So, end goal of cell respiration is to create this energy, usable energy. And so we noticed that in photosynthesis, we were able to make ATP. However, it is very small compared to what you make in cell respiration. And in reality, the ATP made in photosynthesis net goes into cell respiration and is used up to essentially be able to build up that glucose molecule. So for ATP creation with photosynthesis, this is going to be photophosphorylation. Remember, photo is light. And then cellular respiration is going to be oxidative because, well, in cell respiration, oxygen is one of the reactants that goes into it. So there are two types of respirations, aerobic and anaerobic. So remember, aero means oxygen, okay? And the prefix AN means none. Okay. And so when we talk about aerobic versus anaerobic, the big distinction is anaerobic, there is no oxygen. And with aerobic, this is going to require oxygen. Now, we are organisms, us humans, and have the ability to do actually both. Whereas there are some organisms where anaerobic respiration is their primary energy source. Now, anaerobic respiration, I'm going to go ahead and highlight this, is located in the cytoplasm, whereas with aerobic respiration, this is going to happen in the mitochondria. But the thing that's very significant here is the amount of ATP being made. With anaerobic respiration, you only get net 2 ATP, whereas with aerobic respiration, we end up netting with 36 ATP altogether. Now, when we look at the processes, they both use the first step. That we're going to go into that is that is going to be glycolysis. But if oxygen's available, then it goes into the mitochondria, where then it goes through what's called the Krebs cycle, and eventually to the electron transport chain. Without oxygen or anaerobic, we end up getting the lactic acid and alcoholic fermentation. Let's go ahead and take a look at this. So if we look at aerobic respiration, well, we have this first part right here. This glycolysis happens in the cytoplasm, and that is going to be whether with or without oxygen. However, if we're dealing with an organism that has a mitochondria and has oxygen, then you're going to have this additional step where you go through the citric acid cycle and end up going through this electron transport chain to do oxidative phosphorylation to make good amount of ATP. So let's kind of jump into it. So whether you're doing anaerobic or aerobic, the first thing that we're going to be doing is glycolysis, which simply just means splitting sugar. So we're taking that C6H12O6 and essentially breaking it in half. And as a result, that energy that was stored within that carbons of that glucose is used to, number one, create 4 ATP. Now, we had to add ATP to get ATP, so we end up getting a net ATP or a net of 2 ATP. Now, I want to bring up this player right here, NAD8, NAD plus, and NADH. So in cell respiration, we have 2 what's called high energy electron carriers. Now, these electron carriers, what they do is they end up taking on electrons, and that is going to allow them to then carry the energy. And so these little NAD so we're going to be using 2 different types in cell respiration. In photosynthesis, we use NADPH. But with cell respiration, we use NADH and FADH2. Both of these go into breaking down that glucose later on. All right, so a couple of things to also note about this. All organisms do this, okay? So whether you are a bacteria to an animal, everybody does this portion. Now, here's the deal. Glycolysis is good for single-celled organisms like prokaryotes. However, you yourself use copious amounts of ATP. So how do we get more from that? All right. Well, as long as we have the oxygen and we have the organelle mitochondria, we're able to use that energy and break it down even further. So the first thing that happens, again, we go from the cytoplasm into the mitochondria, in particular the membrane of the mitochondria. And as this goes on, what ends up happening is that glucose that came in, that pyruvate from splitting in glycolysis, is essentially broken down piece by piece by piece by piece. At the same time as it's stripping its carbons, well, take a look. It's stripping the carbons and essentially powering these high electron energy carriers. And those are going to go to the electron transport chain here in just a moment. So essentially, we're stripping the glucose of its energy so it can be processed properly. Now, we also do make ATP from it, but a big thing is it only makes 2, okay? The big goal of the Krebs cycle is to create these high energy electron carriers so that we can take that energy that we just stolen from glucose. Now, take note here, as we are breaking down this glucose, we are releasing CO2 as our waste. And as we saw, that was one of our products for cell respiration. But let's go see what happens with the electron transport carriers. So if you're kind of looking at this and you've been following along on our lecture series, you kind of recognize this as being something that we've seen in photosynthesis for the what we saw in the thylakoid and for the light-dependent reaction. And this is set up pretty similar. Big differences would be that we do not have those photosystems in there collecting chlorophyll, but we don't need to. In this case, we already have the high energy in the electrons in these high electron carriers. And so instead of having to excite the electrons, they're already excited and they release them. And when they do, that power, those electrons end up going through these chains of membrane proteins. And what they're doing, very similar to that of what we saw in photosynthesis, uses the energy from those electrons to actively pump hydrogen ions against the concentration gradient. And as we know from prior units, this is a form of active transport. And so as those electrons get stripped and put through, those hydrogens build up. And what do? They travel from high to low, and they go through this ATP synthase. And as a result, they end up making a ton of ATP as a result of it. And so that's going to be that oxidative phosphorylation. That's exactly what's happening here. Back here, we kind of notice that, well, that electrons can't be floating all around. And so here we have oxygen. The oxygen that you're breathing in comes in, snatches those 2 electrons, grabs a couple of hydrogens, and we now have water as a result of it. And so this is the reason why you breathe right here, is for that oxygen to grab those electrons that was stripped from the glucose that was made from photosynthesis. All right, so kind of just go over what we just talked about here. Again, with the electron transport chain, this occurs in the membrane of the mitochondria. This is where it is stripping of their electrons and creates a hydrogen gradient. And that ends up going down through the ATP synthase, making a good amount of ATP as a result of it. And then here it is. If we kind of go, and so we can kind of see overall the basis of what we just talked about. We go from glycolysis to the Krebs cycle to the electron transport chain. All right, so let's go ahead and kind of go through what we know so far. So first step is going to be glycolysis. Is going to be first. Now, we so far has been talking about oxidative phosphorylation with aerobic. And so if there is oxygen available, then it's going to be aerobic respiration, which is going to lead to then oxidative phosphorylation. And that's going to end up giving us 36 ATP. But we need to figure out what happens when there's not oxygen. Back to our flow chart here. So again, If there is no oxygen available, it's going to be the anaerobic respiration, and we are going to have the lactic acid, and we're going to have the alcohol fermentation, and that only results in 2 ATP. All right, and that concludes how we go from the stored energy in glucose to creating the actual cell energy that the cell needs to live. Available. So, for anaerobic respiration, well, unfortunately, fermentation makes no additional ATP. All right. Really, fermentation is just recharging these high electron energy carriers so it can repeat the process of going after another glucose to be able to create energy. And this is why glycolysis is always in continuance when we're doing these kind of things. Now, there's 2 types of fermentation. So for lactic acid fermentation, when they regenerate these NADHs, there's a byproduct of that, and that's the lactic acid. And this lactic acid is seen if you've ever exercised too hard and you have the pain building up in your muscles. Well, that's the result of lactic acid being built up because you did not have enough energy for what you needed, and as a result, your body also switched on to anaerobic respiration with aerobic respiration to try to get additional energy. But as a result, that lactic acid builds up and causes pain as a result of it. But this is also why we have things like yogurt, cheeses, and the such, because that lactic acid allows that milk fat to be curdled. Now, another type of fermentation is alcohol fermentation or ethanol fermentation. This is where, for a lot of yeast especially, and plants, where they break down those glucose, but as an end result, instead of producing lactic acid, they end up producing ethanol. And that ethanol can be used to make beer, wine, etc. But for yeast, that byproduct of that oxygen allows the bread to rise as well. So